Very good morning, dear students, and Assalamu alaikum warahmatullahi wabarakatuh. So we are back after many days. Hope you all are well prepared with the chapters which we have explained, all the other systems of the body. Now we are going to discuss about how does the regeneration or generation of the organisms takes place, the various organisms. How do they reproduce? So we are going to talk about the different type of reproductions in the microorganisms and the plants today. And then this chapter, as it was discussed by some of the students, they wanted it bitwise. So I'm not taking the entire chapter in a I wanted to take the entire chapter at a time, so that you can go through it and you get ample time to go through it. You can rewind and see. But some students are finding it difficult to go back and check it again. So I have split it into phase wise. So today we are going to talk about reproduction in plants and microorganisms. So let me share the screen with you all. And yes. So this is what is we are going to discuss about the reproduction or the generating system. How do individuals give rise to the same, like human being reproduces a human baby, and plant replicates and gives rise to a same shape, size, form, and necessarily the continuous life cycle processing into the offsprings are nothing but the babies, the next generation we are going to discuss about. So how is it happening in is very interesting. And there are two types of ways by which this system's reproductive system works, or the generation of the system works, generating the next generations. And those two types are asexual and sexual reproduction. So what is asexual reproduction? Asexual reproduction is one where only one gamete is involved. There is no male and female gametes. So it's the organisms where there is no differentiation of male and female gamete that reproduces through different techniques like budding, as and spores are used. And you'll come to know about how are the different ways of asexual reproductions and fragmentation, segmentations. There are many ways, very many interesting ways how these organisms are being generated. And then we are going to talk about how is it happening in sexual reproduction. In sexual reproduction, we consider both male and female gametes are involved in giving rise to new generations. Like if you look at the pollination is also, we will be having it now. How does the male gametes from the anther enters, pollen from the anther fertilizes the egg and female gamete and gives rise to a new flower, new plant, new seedlings, new fruit. Fruit is developed, and from the seeds you are again able to replicate and make many more same type of plants. Okay, one mango plant, if you are seeing, and you take a seed, and the similar plant can be produced. So there are different ways by which reproduction happens. So let us start with how are animals, plants, microorganisms. Before that, we will talk about asexual. First, we'll go with the asexual reproduction. So in this asexual reproduction, plants and animals reproduction is necessary life process for the continuous process of production. And if you see that there are male organisms and the female organisms, and the same organisms may reproduce differently in different situations. We can see various ways. for example, if you look at paramecium, it gives rise to more than one of its kinds with a single parent by simply splitting into two. So in paramecium, as I told you, it's a asexual reproduction. So there is no need of male and female gamete. Only there is one parent which is splitting itself, and gives rise to a new generation. If I can use uh, pen, let me see how I do that. Yes. So if you can see my spotlight, you can see that this paramecium is splitting from the center. Okay, from here it is splitting, and now two different individuals, it's giving rise to two different individuals. So that is how, by splitting, this reproduction takes place in paramecium. So, and this is an asexual mode of reproduction. Now, formation of bacterial colony. Let's see how bacteria is formed. If you look at, I sometimes, it amazes me. We are even planning to start, a sort of research based scientific facts with the book of Quran is supposed to be a book of guidance. So if you all have read about Surah Jinn, I sometimes feel that because jinns are something which are invisible to us, and we are not supposed to irritate them or disturb their lives. So sometimes I feel, are these microorganisms which, or the bacteria which we cannot see, like corona is one of the violent jinn that has come up because we were disturbing and polluting their environment. So here, that's why it's a thought. We are going to give you some sort of chapters and exercises where you can do a research and come out with that what exactly it is. So if you look at this experiment, what we can do, and it's very simple, I think each one of you can do it at home. And these activities were even asked in your exams earlier, but I don't know how far these activities, because your 30% of your syllabus is reduced, and this activity is not a lab activity which you are supposed to do in the school. Even at home you can do this. So what are we doing is, we have taken two containers, and in each container we have taken milk. Okay, take one cup of milk in each one of them, and let us have cold milk in one container and warm, lukewarm, not very warm, lukewarm milk in another container. And then what are we doing is, we are adding a spoonful of curd in both the containers. And then what do we observe? Let us see what happens after we add it. And then we are mixing it thoroughly well, stirring and mixing this milk with the curd. And then we are covering both and we'll observe what will happen to it. So what have we done is, cover both the bowls, and now we'll see what happens. Curdling, if curdling has occurred, that means the bacterial colony. We have got a lot of bacteria which we cannot see in the environment because they are microscopic. We cannot see it through our naked eyes. So what is happening is, when you have covered the milk, already this bacteria has entered in the milk bowl. And this bacteria curdling indicates that there is an increase in number of bacteria. Note the time taken for the formation of curd in both the bowls. If you look at the formation of milk in both the bowls, what do you observe? You have observed that there is the lukewarm milk has curdled. Does it take same time? You have to do this experiment and see. So you will find that the lukewarm milk curdles faster than the cold milk. So what is the reason behind it? Why is it happening? So there is no formation of curd in the bowl that contains lukewarm milk because it takes 5 to 6 hours. In the second bowl, in which there is a cold milk is present, no curdling took place. So why did the lukewarm milk took 5 to 6 hours, and the cold milk it could not get curdled so quickly? So that is the reason where that was a congenial environment for the bacteria to grow and curdle into the lukewarm milk. So what happens is, within 30 times the size of the bacterial colonies are indicated. So the bacterial colonies indicates that how fast bacteria are growing in a lukewarm milk. And similarly, so this is how it has happened. Lactobacillus bacteria, which is supposed to be one of the very good nutritious bacteria that help in the curdling of your milk, and curd is consumed by us as a nutritive food. So now let us see what is a process, asexual process of budding. What happens in budding? A small bud is there which grows from nearly the same identical copy of a parent. It's just same like its parent, and that same bud, it grows back like its parent. And in this, if you just see, it is how is the cycle of the budding. G1 is a gap, and S is the DNA synthesis, G2 is gap 2, and M is mitosis. Mitosis and meiosis division you have already studied. You know how mitosis and meiosis division takes place. So this DNA interface, this is the whole cycle or the whole process of budding that takes place in asexual reproduction in plants. So just go back and revise your cell and its organelles. Understanding cell and its organelles is very important, because at every stage, every time when you are reading science, a single cell, because human beings are made up of billions and trillions of cells, and these cells having same function together forms a tissue, and a group of tissue having same function forms an organ, and we know that organs forms a system, and all the systems together. Next chapter you'll be having coordination. How do they coordinate, and the functioning, the body functioning takes place. So you know that a cell consists of a cell wall, centrosome, nuclear membrane, and cytokinesis is a process by which cytoplasm splits and divides it into two parts. And this entire cycle, how it is happening. If you look at how the centrosome is duplicating, and through the centrosome, another gene is emerging out, and then the centrosome separates, forming a nuclear material is migrating, and then a spindle like. During your mitotic division you see all these changes that takes place, and finally a daughter cell is produced through this nuclear spindle like elongation takes place into it, and then the nuclear material very equally divides. You see that if it is not getting divided equally, there is a lot of problems we find into a daughter nuclei. So that is how through mitosis division budding takes place, and a new daughter cell is formed through mitotic division, and a new organism is generates. And what happens in fragmentation? So in fragmentation we see that the plant, a small plant is been. So how many types we have seen by now? In paramecium you have seen that simply the body is splitting, through splitting the body gets splitted by a single parent, and then it divides and forms daughter parameciums. And then you have seen how it happens in buddings. During budding time, through the mitosis and the mitotic divisions. Mitotic division does not take place, only mitosis, because as you know that mitosis takes place when it comes to sexual reproduction. And here we are talking about asexual reproduction. So through a process of mitosis, you know the entire cycle of mitosis: anaphase, metaphase, telophase. So in the last telophase stage, how a spindle is getting divided and a daughter cell is formed. And now let us see what happens in fragmentation. So if you look at some of the organisms, what happened, a part of their body, like fragments, like your fingers, if you just see a finger like projections, we have got these differences. So this gets splitted from the organism, and that falls down and grows into a new organism. You can see how it is happening in the figure also. So this happens only in simplest form of organisms. If you look at the tapeworms, molds, generally we see the molds, and you have got most of the mold activities which you have already done. Bread molds in your sixth standard, you had an activity where you have seen how bread molds are formed. Molds are also being, the reproduction of the generating system that is used in molds is also through fragmentation, as well as lichens and spirogyra. They all, the one which we are watching here is the spirogyra organism, which grows through this process of fragmentation in this manner which we are seeing here. Okay, so now if you look at flatworms are again, a part of it, from the a segment of it is getting separated from the parent body, and it falls down and develops into a new tapeworm, the new tapeworm. And in the molds also we see how it is. Molds are formed not only on the bread, even at home. If you are keeping fruits or vegetables which are not refrigerated and which are kept outside, we are giving ample opportunity for these molds to develop. As you have seen in the experiment of milk, when the milk was lukewarm, we could see the colonies of bacteria, lactobacillus bacteria, that is called as lactobacillus bacteria colonies forming in lukewarm milk, whereas in cold it was not there. So whenever the temperature is very cold, these molds or the bacteria cannot generate there. So temperature is a very important factor or a variable that is important during your experiments. You have to see that. So when you're keeping it outside, you can see these molds forming. And how do these molds generate? So through asexual reproduction, these molds are multiplying in millions and millions, and that is how it is, the entire fruit is getting destroyed. And even in lichens, you see these lichens on most of the plants, on the wood also at home, some certain times you can observe these lichens. And spirogyra. So this is how it is developed. And you have got, I think, a simple activity there where you can check, you can have the blood mold and bread mold, and see the rhizopus in the lab. These may also reproduce sexually. There is a possibility that not only asexual reproduction is taking place, because the way they are multiplying, they may even be reproducing sexually. So now fragmentation is a common mode of reproduction in large fungi and many plants. If you can see mushrooms. So in mushrooms also we see that they are getting fragmented into different areas. And these fragmented uh, organisms are giving rise to, one sec, let me check how is it written here. let's resume back to a share screen. So you can see how this is asexual reproduction is taking place into these plants. How is spore formation taking place? So if you look at it, spore formation is a method of asexual reproduction which occurs through microscopic reproductive units called spores. So these spores you can see only through microscope. And in this, you know what happens when these spores they are actually stored into sporangium, and they are called as sporangiophore. And when this sporangiophore burst out, many, we cannot even count the millions and millions of spores are thrown out into the atmosphere, and wherever it falls, it again starts growing into a new it starts growing into a new baby organisms, and this is purely a asexual sort of reproduction. So we can see in the rhizopus is a very common example of which we can even see it in your lab activities, how this is happening through, and we call it as its roots that falls, it is called as mycelium, and this is stolons. These are the mating type 1 and mating type 2. So both asexual reproduction is through asexual reproduction it occurs, and we can observe it through a microscope. How is it happening? Okay, the whole process. We'll do a lab activity also here only, and check. You can see it very clearly how it is happening. So you can see that most of the fungi of rhizopus or mucor, bacteria, non-flowering plants such as ferns, mosses, they reproduce by the spore formation method. So this is how you have seen how the spores, sporangia, once it matures and bursts out, we see a lot of spores, millions and trillions of spores that falls down, and out of those spores again the new organisms generates. New rhizopus generates. So this is how it happens in, if you look at this bacteria and non-flowering plants, how they are reproducing through this spore formation. And we can even use artificial way of propagation of this spore formation. And most of the fungi like rhizopus, mucor, bacteria, non-flowering plants such as ferns and mosses, they reproduce by this method. So you can see how the fern plant looks like, and mosses plants, ferns are. So they have got a complete cycle through which the fertilization takes place and a new gametophyte. Let me check it out. I don't think that this entire cycle is there for you. If it is there, I'll explain. Otherwise, I'll not go into the details which will confuse you. Oh, do you have this cycle of Hydrangea? Let me check. Okay, fragmentation, budding. Okay, it is not explained in detail, so don't go for it through mitotic and meiotic division, because certain times sexual and asexual both the ways of fertilization takes place, and that's how this they generate or reproduce. So you can see that a fungi produce in some specialized structure called sporangia, and which burst and spread the spores in the air. So through the air or the wind, the pollination takes place in the flowering plant. Similarly, these spores, when they land on the food or soil, and whenever there is a favorable condition, as you know, the temperature, water, soil, what all are the favorable conditions, or the food item on which they falls, which is favorable for them, they start reproducing into a new plant over there. And this is completely a pure asexual reproduction. And if you look at the fern plants, it's also very interesting, and there are so many fern plants near our school. You please go and check at the back side of the leaves. If you turn the leaves and see, you can see a lot of sporophylls and a cluster of sporangia on the lower side of it. This yellow colored which you see here, these are all the sporangia or the spores. So you can just imagine from one leaf of the fern plant how many of them can fall down, and because of the wind they are very light. They are being carried away to the far off places, and where a new organism generates, or a new plant, fern planted takes place. This is the fern plant, and it is very quick and easily we find it getting these plants are very easy to grow. And these are all non-flowering plants, and the leaves like sporophyll bears cluster of sporangia. You can see that these all at the back side of it, of the leaf, fern leaf, what you see is nothing but sporangia. So this is how through this artificial reproduction can also take place in these non-flowering plants. You can see these are the sporangia on the lower side of the leaf, and these sporangia, they produce spores which produce the new plants when it falls on the ground. You can see how it is falling on the ground and then producing a new one. So let us do a lab activity to observe how a rhizopus or a common mold is formed. So what is the aim of our activity is to grow and examine rhizopus. We are going to see how rhizopus is there. And as you all know, we need some materials. So there are simple materials which we need. We need bread, we need one plastic bag, eyedropper, cover slip, plain glass slide, disposable gloves. Gloves are very important. Don't just use them with your naked hands, because if you have got allergies, you may suffer. So when you're doing this experiment, take precautions that you use gloves, and immediately you clean and wash your hands also. Like, it is not that this corona, now at the corona time we are taking all these precautions, whereas these bacteria were present in the environment from a very long time. So every time it's important that you clean and wash your hands. So this is a eyedropper and a cover slip, and you see a disposable gloves, and microscope is very important under which you are going to observe. So how are we growing molds? You see, if you take a piece of bread, and then what are you doing is you are going to preserve that is the preservative free, and leave it in the open for some time, and sprinkle some water on that bread and put it inside a plastic bag, and then you tightly pack that plastic bag and seal it. And after after few days, you just add dry it out. Okay, check on a piece, and after a few days, what are you doing is you are checking it out. What do you see? That there are some black mosses, the gray green fine dot structure. They are sometimes black, sometimes green. Green is poisonous. Generally, to the children when they are eating bread, we'll say that don't, when once it is, there is something black or green on it, it is poison. But actually this is rhizopus, and of course it becomes poisonous also for you to eat. There are a lot of food poisoning in these molded breads. If the people are eating, they suffer with food poisoning. So the black dotted structure is called as this bread mold. So now we are going to observe this mold under a microscope. So a good sample of mold within 2 weeks you can collect. Take it, put it into a slide, add a drop of water, cover it with a slip, and then take a tissue paper and clean all the surroundings and press the cover slip properly, and then observe it under the microscope. So what you see under the microscope, he's even showing you what is there under it. So you can see this rhizopus mold very clearly under microscope. Observe it again. So this we can observe under this microscope. So this is what is the procedure to make a slide. So how are you going to make a slide? First, drop some water using an eyedropper on your slide with the tip of it. Okay, and then use a toothpick. Never touch it with your fingers or hands. Use a toothpick or some instruments through which you can easily spread this mold on the slide, and then cover it the slide using a cover slip and fix it properly. Clean it with and make sure there are no air bubbles. The precautions you have to take is the specimen should not have air bubbles, otherwise you cannot observe it under the microscope. So now with a blotting paper, you can cover or clean all the edges of it, and keep this slide under the microscope and observe. So what do you see under the microscope? Your observations will be this. Okay. So it contains thread-like projections, and these projections are called hyphae. And these thin knob-like structures, they are called sporangia, which contains spores inside it. And these spores are used for regeneration. And important thing is what precautions you are going to take. The precautions are very important because these molds are you may have skin allergy or other asthmas. The people having asthma or anything, it's not safe for them. So thoroughly after this experiment, make sure that you use a mask also when you're doing an experiment, and wash your hands thoroughly. And similarly, you can even observe, it is very easy, anywhere on the fern plant you can see this sporangia, the cluster of these dots, like these are nothing but the spores, which you can even see them with your naked eyes. You can just remove it with the needle also and check this spores, or with a magnifying glass. So now let us go to the sexual reproduction. In by next this thing next video, I'll be sharing with you with the sexual reproduction. But before that, I'm giving you time till Monday. By Monday, you all have to either mail it or on WhatsApp. You have to message it that yes, you have understood the concept thoroughly well, so that I can go for the next video. So till then, take care, be safe, and see you all back on Monday.